Since I'm also co-chairing the next section, I'll tell you we'll get caught up. So I'm going to get started in the interest of time here. What I want to talk about today is something which is my favorite topic, which is asymmetric self-renewal as it relates to adult tissue stem cells. And in the context of this meeting, I want to talk about how it's a fundamental property of these adult tissue stem cells. And it's also a very important principle for thinking about how we do the medical engineering of tissue stem cells. So I just want to start off with this conflict of interest disclosure statement, because Asymmetrics is in fact a for-profit company, and many of the technologies that I'm going to talk about today are technologies that we are currently trying to commercialize. So there are three main areas of stem cell medical engineering that I want to address today with respect to asymmetric self-renewal. One of those is transplantation medicine, which we started talking about this morning in my keynote address. I want to spend some time talking about tissue engineering and also drug discovery as it relates to stem cell medical engineering. So just to review again, stem cell medical engineering from the standpoint of transplantation medicine, the idea is that we have individuals who have a problem, in this case, the pancreas. What we like to be able to do is to replace it with cells, or in this case, cells from a person who's potentially could be a living donor, to improve the state of health of this individual. We want to keep in mind that some of these transplants are also from deceased individuals as well. It's an important part of the donors for these for transplantation medicine. In terms of our second category of tissue engineering, comes in a lot of different forms. One of them is taking cells from an organ and in culture expanding those stem cells so that we have more of them to provide to more individuals for transplantation medicine. The same strategy of expanding cells can be used to create engineered devices that might be more efficient for returning to patients, may solve problems like immune difficulties. And again, the idea here is that by developing these engineered systems, one might be able to help more patients. And by drug discovery, what I mean is, again, taking tissue stem cells and having them in form and in number to allow us to either do engineered systems for screening for drug effects, for screening for drug toxicity, ultimately to develop pharmacological agents that can affect the same types of changes in terms of patients as the cells have. So these are sort of the three areas of stem cell medical engineering in a broad category. And what I want to turn to now is talking about why asymmetric self-renewal is important for each of these. And to get us to that point, I need to first address the cells that we have currently available for doing stem cell medical engineering. There are three major types. There are some other ones that aren't talked about as much, but embryonic stem cells, induced pluripotent stem cells, and adult tissue stem cells. Now, in terms of source, and each one of these cell types used for medical engineering has special properties and also some important challenges. Now, one of the challenges for embryonic stem cells has to do with their source. They're from donated human embryos. Those embryos have to be destroyed to create the embryonic stem cells, and there are ethical objections to that because of the destruction of human life. Some of the special features of embryonic cells is that they can undergo multiple types of cell differentiation. This is both a plus and a minus. The plus side is you have one type of cell that can give rise to many different types of tissues. The downside is that typically when we're doing cell therapy, we're only really interested in transplanting one type of tissue, and these other forms of differentiation lead to a need to be able to purify out the specific cells that are needed for a given transplantation. There's also the problem of getting rid of any undifferentiated cells, which are tumor-forming in mature tissues. Now, one of the things that we've now learned over time that wasn't appreciated 10 years ago is that we don't get the desired mature differentiation from these cells. In most cases, these cells differentiate into cells that are closer in development to them. They're early fetal differentiation. There's a question whether the fetal phenotypes are sufficient for drug development for adults and children, and for transplantation into adults and children to achieve the same types of functions as adult tissues. And I've alluded to the inherent genetic mutations that are part and parcel with making embryonic stem cells. Those mutations are problematic in terms of forming tumors in mature tissues, and they may also compromise the differentiated cells that are produced from embryonic stem cells. Now, everything I said here for embryonic stem cells apply to induced pluripotent stem cells as well. One of the differences, though, is that these cells can be produced from individuals who have disease, and so one can get disease models with induced pluripotent stem cells that are not possible with the embryonic cells. But all the other caveats still apply, except for the ethical one, because these cells are derived from adult donor tissues, and consent can be done to do that. Now, adult tissue stem cells are found in both adults and children, and they can be sourced by appropriate consent. The challenges here is, as I mentioned earlier this morning, these cells, generally speaking, are difficult to expand. I'll talk a little bit about that. There is some progress. Our company has a technology called SAC, for suppression of asymmetric cell kinetics, which is providing some relief from that problem. One of the big advantages in terms of being able to make mature tissues, these cells are tissue-specific in their differentiation. Stem cells from liver make liver stem cells. They do it spontaneously in many cases, in the sense of generating mature cells as opposed to immature cells. And the defining and unique property of adult stem cells is what I want to talk about today, is asymmetric self-renewal, which is distinct from both of these other cell types. So there's a very common, pervasive stem cell definition. Most of us know it. We say stem cells, you're a stem cell if you can self-renew and differentiate. Self-renewal in this case, I'm showing a symmetric self-renewal where stem cells can divide, make two, essentially the same cells as the dividing cell, or undergo differentiation where division gives different types of cells. So this definition is not quite adequate for defining adult tissue stem cells. And the reason for that is because something happened with this slide. Yeah. The reason for that is that we need to say a little bit more. So the mnemonic, the thing which names the adult tissue stem cell, is this ability to undergo asymmetric self-renewal, defined by self-renewal and differentiation, two additional words, continuously and simultaneously. And that's what's diagrammed here, that these cells undergo asymmetric self-renewal, which means they can divide, give rise to a committed differentiating lineage, and they maintain their stemless state. And we'll talk about in a couple of moments about the manner in which this is achieved. But that's the defining feature. And that's what we want to talk about, because very often this is overlooked, and yet it's very important in terms of thinking about engineering these cells for medicine. I want to make a distinction here, because there's asymmetric division, which occurs during embryonic development, occurs in the development of mature tissues even as we sit here now. So there's no question about asymmetric divisions. But what we're talking about here are these asymmetric divisions that are different in terms of asymmetric self-renewal. So during development, there are asymmetric divisions where the two sisters are different than the parent. But importantly, both sisters differ from the parent in many cases. And these divisions are limited. They occur during a certain window of a developmental program. They don't occur for the entire lifespan in those, unless those programs are still running. In terms of asymmetric self-renewal, again, the two sisters, this cell and that cell, are unequal. But one of the sisters is different. One of the sisters is exactly the same as the parent. That's the idea. And this is continuous over the lifespan of the organism in which these cells reside. So I just want to make that distinction. I want to give you one example of this. There are many, many more. But this is the crypt villus unit of the small intestines. How do we know stem cells exist in these units and we can't see them or we can't count them? It's because of the turnover. This system looks static, but in fact it's constantly turning over. Cells are being produced here by asymmetric self-renewal of tissue stem cells. They're giving rise to the cells that undergo differentiation, and ultimately out here those cells undergo cell death. That process requires that these cells divide and make something different and don't forget who they are. They retain the program continuously throughout life. That's the asymmetric self-renewal, and there are many other examples of it in mammalian tissues. So what I want to turn to now is how the asymmetric self-renewal is a factor in each of these different types of stem cell medical engineering, starting with transplantation medicine. So the top here, which I wish I could make this bolder, I want to talk about why the pluripotent cells, embryonic and induced pluripotent cells, are not adequate for transplantation medicine. And the reason is because they have self-renewal and differentiation, but it's actually self-renewal or differentiation. So when embryonic cells or induced pluripotent stem cells are proliferating, they maintain their stemless state, but when you tell those cells to differentiate, they stop being stem cells. They are converted from this form to these phenotypes here, which are non-stem in nature. Right? So that's how we achieve the production and making large quantities of the differentiating cells. But this is a problem for doing transplantation medicine. In contrast, adult tissue stem cells, which have asymmetric self-renewal, do self-renewal and differentiation continuously and simultaneously. Now, there's a lot of debate. It's been going on for 50 years about how this is accomplished. Maybe even one of these isn't the right way. But they're two sort of competing notions. One is that the cells do deterministic division. An individual cell divides, reproduces itself in something that's going to be the progenitor for a committed lineage. That's shown here. But the same thing can be achieved by having small populations of stem cells where some are undergoing these symmetric divisions, making more stem cells, while others are undergoing differentiation in a balanced fashion. The net is the same idea. We have systems that maintain the program for the tissue while producing the differentiating tissue. Now, that's really important if you want to do transplantation medicine and have long-term repair. This is a schematic here of a quote-unquote normal tissue with asymmetric self-renewal divisions taking place, like our skin, our hair, our liver. Damaged or diseased tissue, we lose these units somehow. How do we repair them? Not so well if we use the pluripotent approach, because we can take pluripotent cells and generate these differentiated cells and put them into tissues. But depending on the turnover rate of that tissue, this is not going to be a long live repair, because these cells cannot recapitulate the entire tissue. It's just that basic. So the repair is going to be short-lived, and if you happen to put some of the actual pluripotent stem cells into the tissue, tumors arise because of it. In contrast with these tissue stem cells, those stem cells which have asymmetric self-renewal, if you put them into tissues, they will over time reconstitute this tissue, and they will do it continuously and for long periods. This is classic bone marrow transplantation with hematopoietic stem cells. So this is an important principle for transplantation medicine for long-term replacement and restoration of tissues. I want to turn now to asymmetric self-renewal in terms of tissue engineering. Why is it an important principle there? I'm going to focus on, there are many aspects of tissue engineering as I showed you. One of them is having stem cell biomarkers. Right? So markers that tell us we're looking at stem cells specifically that would allow us to count them don't really exist now for most types of stem cells. This asterisk is for me to remind me to say that this is also important for transplantation medicine, where we do stem cell treatment not knowing the stem cell number, and for drug discovery, as I'll mention at the very end of my talk. So here's this is the current dilemma of biomarkers for tissue stem cells. I'm going to just sort of say there aren't any, in the sense that they are specific for stem cells. This is best illustrated where our best biomarkers are for the hematopoietic stem cells. The reason this situation exists is because the cells are scarce, which means it's hard to get enough of these cells to actually be able to do a good job of finding specific biomarkers. We don't have good quantitative assays, so our assays aren't quantitative. It makes it difficult, again, to know the number of cells and be able to come up with better biomarkers for them. This is illustrated here for a very well-characterized clinical population that contains hematopoietic stem cells. There's no doubt there's hematopoietic stem cells because you can do two things with them. You can rescue patients who've had their bone marrow ablated, and you can also show that in animals these cells would in fact give rise to all of the human hematopoietic system cells. However, for every 10,000 mobilized peripheral blood cells, for the one hematopoietic stem cell that's in that preparation, which is a low fraction, there are about 100 CD34 positive cells, and there are about 50 CD133 cells, because they share the same marker as the hematopoietic stem cell which gave rise to them. And because they're so much more predominant, this ends up not being it's a great biomarker to have, but you can't use it to quantify stem cell number. right? And that's challenging for lots of things we like to do in stem cell engineering, not being able to quantify these cells. So one of the things that we've been working on is concepts for coming up with more specific biomarkers. I just want to mention one approach briefly. We've been looking for genes which are strictly associated with asymmetric self-renewal. The idea is that asymmetric self-renewal is much more specific for many types of tissue stem cells. If you find markers that are specific for the cell undergoing asymmetric self-renewal, you may have a better marker, a more specific stem cell marker. I just want to give you one example of one of our candidates. We call these asymmetric self-renewal associated genes. This is a protein that's known as CXCR6. We're looking at a system that allows us to tell the difference between symmetric and asymmetric divisions by stem cells. There are two cells here. They're recently divided. Here are their nuclei. We can score this as a symmetric division because they both show the cyclin A, which means they're both going through the cell cycle. Their CXCR6 expression level is more general in the cell and low. This is an asymmetric division where one of the daughter cells is actually going to undergo differentiation. The other one is an asymmetric self-renewing stem cell. And what you see is that CXCR6 is very strongly expressed in the nucleus of that cell. So it'd be great if this was even on the surface, but the basic idea is that we want to find, and we've been characterizing these biomarkers which are specific for asymmetric self-renewal. We think they're going to be the best ones of getting us to the point where we can actually quantify stem cell numbers specifically. I want to close with drug discovery. It's one of the most exciting things happening in our company right now. And the challenge for drug discovery, where you want to learn things about the responses of stem cells to maybe activating agents, or the responses of stem cells to drugs that are toxic to them, which make for very bad drugs, is that we can't identify them exclusively, as I just mentioned, because of the biomarker problem, which means we can't count them. So how do we assay for effects on tissue stem cells when we lack these two abilities? Well, we take a computer simulation approach. It's a little bit far from the problem, but the basic idea is that if we have a good computer model, that we can infer what's going on with stem cells because of their asymmetric self-renewal output. And that's based on the idea that when you take primary human cells and put them in culture for long periods, the cell that's most important for the longevity of that culture is a cell that has a longevity for growth capacity, which is the stem cell. So some of you may be familiar with these types of plots where you look in time, in culture, serial passaging, primary human tissues, and you follow the cumulative population doublings. These are characteristic curves. There are no exceptions for normal human tissues. You hardly ever get to make categorical statements in biology, and one should be careful when you do. But I like saying that one. What you see for any human tissue, if you put it in culture and you serially passage it, so you let the cells fill up the vessel, you take a fraction, you transfer it, and let it happen over and over again. You keep track of doublings. Doublings go up with time, but eventually what happens is every culture is going to stop dividing. The only exception actually is a p53 mutant, half heterozygous mutants, which in fact don't do this. But most of them do. So we say this is based on asymmetric self-renewal, based on this model shown here. This is a stem cell. There are about 4 different things it can do. In culture, it can die, it can not divide at all, it can divide symmetrically, as we've talked about, it can divide asymmetrically. And when it divides asymmetrically, it gives rise to these sort of tissue units where cells undergo transient amplifications, but ultimately they all stop dividing. And that arrest that we see with time, we propose, is due to these cells which are low in number and which don't divide this way nearly as much as they divide in this amount. This is the default. They get diluted out by their own differentiated progeny. Over time, you just lose them by dilution. Now, in computer simulation and modeling in general, the likelihood that what I just showed you is true depends on analyzing data. So what we've done is we've done an experiment, classical sort of serial passaging. So we have data, and we fit our computer simulation to it. And the first question is, how well can we fit the data with these ideas? And we do very well. So these are data here. There are actually 6 individual cultures followed serially over time. They show the increase in population doubling, but they all stop dividing at some point. This dark line here is the average of them. And you can see they stop at different times and they stop at different levels. That's an inherent variance that reflects, in fact, the stem cell behavior and a lot of other things. This is our simulation of that data. And you see we do a very good job not only modeling the magnitude, but more importantly, the variance that's there. And when we're able to get good fits and simulations to data like this, we can then go in and deconstruct the model and ask specific questions about the components, such as what's happening to stem cell number versus time. And that's shown here, where this is actually number of stem cells in these cultures versus time. Is a simulation, is our prediction. And what you see is these cells increase, stem cell number increases as the culture grows. The sudden drop is because we split the culture. And then we get growth again, but you notice the growth doesn't reach nearly the same level. It's because of the dilution that's taking place. There are actually fewer stem cells being transplanted each time because of all of the other cells they're competing with. So that's where we are with this. We have a way of estimating stem cell number, any tissue that we can grow in culture. And we're currently talking to pharmaceutical companies about using this to either look for agents that can activate stem cells for growth, so stem cell number should increase in these simulations based on the data growth that we follow, or in cases where there's toxicity, which we're very interested in, finding agents that actually decrease stem cell number, change the way stem cells divide. So in summary, what I've shared with you is our ideas that asymmetric self-renewal is very important for stem cell medical engineering, but it's often overlooked. And since it's a fundamental of the nature of how our tissues work, it's going to be very important and an important principle in many aspects of stem cell medical engineering. And what I suggest today is that we need to think about incorporating this more into our practice, because that should accelerate our approach to better treatments for patients. Just want to mention my collaborator, AlphaStar Corporation, for the computer simulation work that I've done. And I'm going to stop there, and I'm going to be around for the entire meeting because I know we need to move ahead now. Thank you for your attention.